आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़, की आवाज़।
Vigyan Ki Awaaz podcast series aims to popularize new ideas of science and technology, which highlight STEM, science, technology, engineering, mathematics, and medicine. This brings to the surface the efforts and researches of Indian scientists, which is desired to bring a change in community learning and teaching, access to knowledge of resources, and social paradigms. Our goal is to spread the name of indigenous Indian research stories and their masters among the masses of society in a very easy manner. Bose Institute was set up in 1917 by Sir Jagadish Chandra Bose, the founder of modern science in the Indian subcontinent. It is Asia's first modern research center devoted to interdisciplinary research and bears a century-old tradition. The institute was founded as not merely a laboratory but a temple, with its principal aims being the advancement of science and diffusion of knowledge. Swami Vivekananda, Sister Nivedita, whose influence is reflected in the institute's emblem of Vajra, was significant in the setting up of the institute, and Rabindranath Tagore were among those who had consistently inspired and supported Jagadish Chandra Bose in his endeavors. Ever since the establishment of the Department of Science and Technology, Government of India in May 1971, Bose Institute has been an autonomous grant-in-aid institution of DST. Over the last century, the institute has endlessly strived to preserve the legacy of its illustrious founder. In this episode of our podcast, we have Professor Suman Banik, who is a professor in the Department of Chemical Sciences in Bose Institute. We start this podcast with an introduction to his work, and then Professor discusses in detail about his present work and its significance.
I'm Suman Banik. I'm in the Department of Chemical Sciences, and I'm a professor right now. And my basic education is in chemistry, BS and MS in chemistry with a specialization in physical chemistry. And then I did my PhD in Indian Association for the Cultivation of Science in the Physical Chemistry Department. My research topic was reaction kinetics, and it was a theoretical research. And so after the end of my PhD, I was looking for something new. And so I came across a very nice book of mathematical biology by Jim Murray. And after reading that book, I just got interested in biological system. And I saw that the principles of reaction mechanisms could be applied to biological systems as well. Because in chemistry, what you see that two molecules or several molecules interact with each other in a solution or in gas phase, mostly in solution. So what happens in biological system, the same thing, but in a bigger scale. There are larger macromolecules, they are interacting with each other, and they control certain mechanism, which now is a part of school textbook, which we know as a gene regulation. So everybody in the community, or the even common people of our country knows about the gene mechanism, gene regulation, all these things, because these are taught at the school level right now, at the secondary level, or in the class 2 level. So I saw that I could apply those ideas of chemical interactions in biological system. And after then getting my PhD degree, I moved to abroad and did some postdoc, one in Max Planck Institute for Physics of Complex System in the biological physics department, and then a second one in Nordita, Nordic Institute for Theoretical Physics, also works on some topic in biological system. And then I moved to USA and joined the biological science department and the physics department. And there actually I got a full flavor of the implication or the application of chemical principles in biological system. So there I started working on cell cycle mechanism. And cell cycle mechanism is also a part of textbook material now. Then after finishing my postdoctoral research, I came back to India in 2000. At the end of 2008, I joined Bose Institute and joined as in the systems biology program. So the systems biology program was looking for, it was an initiative that will bring in the theoretical people and experimental people together to understand biological phenomena. And one of my research actually I worked on was trying to understand a molecular mechanism or molecular switch in Bordetella pertussis. So Bordetella pertussis is the causative bacteria for the disease pertussis or whooping cough in babies, small babies. And it's a big challenge for developing countries like ours and also developed countries like USA. So what we did, we using the principles of chemical reaction mechanism or chemical kinetics, we developed a model which could explain a temperature sensitive molecular switch. So what happens actually when Bordetella is in the surrounding or in the air, it doesn't do anything. It goes inside the body through our respiratory system and it starts anchoring in our lung. And then at the body temperature, 37 degrees centigrade, it starts expressing its virulent genes. And that causes the disease. So we showed how using the principle of chemical kinetics mechanism and some dynamical systems principle of physics, how a molecular switch is working, a temperature-mediated molecular switch is working in Bordetella pertussis. And then we started working on biological phenomena in looking for principles in single-cell environment. So why are we interested in single-cell environment? This simple system, these bacteria are basically, they are sometime our friend or sometime our enemy. When it's in the gut system, it helps us to digest our food. But when it goes inside our body to our respiratory system, it does a lot of trouble. So, but why single cell? Because it has been shown experimentally that each of these cells in a colony, in a population, they behave differently during the gene expression mechanism. So, and it is connected to a nice, interesting phenomena. It is a nice but deadly phenomena called quorum sensing. So we all know the cholera, about Asiatic cholera, which was at some point of time, it was a big challenge for our country. And it's coming back again. So what happens in cholera is that individual cells of choleras, they talk with each other and they communicate with each other. What they do, they send some signaling molecules in the solution, and through these signaling molecules, they know what is the population size. So they basically look for the quorum. Okay, we know that in a meeting, when we have a lot of members in the meeting, we always look for quorum. Unless without the quorum, we cannot go for the meeting. The proceedings cannot go further. So what happens in cholera as well, the cholera individual bacterium, they try to grow, the population becomes larger, and the signaling molecules they send, it gives a signal within the cell. It has a pathway, some signaling pathways, so where they tell, well, now we are ready to release the toxin. So if you read all these fictions and these, that somebody ate some food and then all on a sudden they start vomiting and there's this diarrhea and they become ill and eventually they die. They lose so much of body fluid. So we try to look at the quorum sensing mechanism. So together with one of my colleagues in the biological science department, we are trying to understand that how to control this quorum sensing behavior in bacteria. So there has been report that some inhibitor molecules are available, which are sometimes they are natural, sometimes these molecules are synthetically made. So we are looking for this quorum sensing control by using these artificial molecules. So what we will do is we will start doing experiment first, and then we will do our theoretical modeling. And from the theoretical model, we will predict something. Then these predictions, again, we will go back to the experiment so that it can be verified. So it's a back and forth mechanism. That's how people work together in collaboration when they do combined theory and experiment. Okay, so now what we do is a lot of people are now in this community. Now they are doing theoretical science, and they are collaborating with experimental guys. So, and they are trying to understand biological phenomena. So in our country, this interdisciplinary research was when I started this kind of research, it was very nominal. Very few places used to work in that way. Now it has increased. A lot of new people has joined and they are doing interdisciplinary science. So they are using all the tools of natural science, physics, chemistry, mathematics, biology. They are putting all these ideas together to understand complex biological phenomena. As I said earlier, this biological system is a very complex thing, and all this mechanism, these big macromolecules, these molecules are interacting with each other in a soup. Okay, so this is one aspect of research. So another aspect of research, as I said, the single cell, but each single cell has this, as I said, this phenotypic heterogeneity. So what is phenotypic heterogeneity? So some cells, as I said, the gene regulation mechanism takes place in a soup, in a turbulent soup, I would say. So in a turbulent soup, so they use all the cells do not express their gene at the same time. As if you have a couple of babies in your hospital, few babies are crying and most of the babies are sleeping. So when it's crying babies, you can map it into a cell, a gene which is getting expressed, and the sleeping babies where the genes are off. Okay, so now if you look at the data, if you look at the experimental data, single cell expression data, you will always find a distribution. Okay, so there are methods to find out this distribution and quantify these distributions. Now the question is what these distributions are telling us. These distributions are telling us that these mechanism of this gene expression mechanisms are not deterministic but probabilistic in nature. Now this comes this concept of probability, And this notion of probability brings in the idea of information processing. Now, why this idea of information processing is coming in? Because a living species, well, we are looking at bacteria, it faces different signals from the environment. And when they face these signals, they process these signals and to downregulate, express or repress certain genes responsible for their functions. As in quorum sensing, as in cholera, I said, is release of toxin genes. Now, this information processing is vital. If we can measure how much information is processed and how this information processing takes place, we can control them. We can manipulate them experimentally. In the lab, we can do genetic engineering in the whole circuit of signal transduction, and we can make predictions that how to control this disease. But we don't want to eradicate them from the system because bacteria are there long before human came into Earth. Okay, so these ideas can be easily translated experimentally and to the common people, so people are aware. I mean, I'm not sure whether people really know there's the cholera bacteria, the bacteria which are causative agent of cholera, they talk with each other. They talk with each other in their own way, as I said. And so it kind of creates an awareness in the system. And well, industrially, I mean, I don't really see right now the direct implication, but maybe in future there will be some follow-up research which will collaborate, which would connect the industrial research and industrial application and the basic science research.
We are immensely thankful to Sir for such an enriching discussion.